Hello, dear students. So now today we would move ahead in this second chapter of class 10 science, that is acids, bases and salts. So today we are going to discuss about the types of indicator. In the previous video I have told you about some commonly used indicators that we are using commonly. That means methyl orange, phenolphthalein and the litmus. So these are the three commonly used indicators. So now today we are going to discuss about the types of indicators. So if you see, generally we have three types of indicators. Okay, some are natural indicators, some are synthetic indicators and some are olfactory indicators. So one by one we would discuss all these. So first we would discuss about the natural indicators. Okay, natural indicators means the indicators which are found there in the nature, means which we extract from the natural things. Okay, which we extract from the natural things, so that we call as the natural indicators. Okay, so likewise in the previous case I have just told you about three commonly used indicator. Most commonly we are using three indicators. Okay, like methyl orange, phenolphthalein and then we have the litmus solution. So the litmus is the natural indicator here, because we just extract this litmus from the plant that is also known as the lichen. Okay, so we extract this from the lichen plant. So let's discuss in detail this natural indicators. Before this, you note it down. So here I have made a list. I have just, or these are the natural indicators. All these are the natural indicators. So first one is litmus solution. Then you have the turmeric solution, that we are also using turmeric powder in our cooking items. Okay. Then you have beetroot juice. Then you have red cabbage juice, also means the extract from the red cabbage. Okay. Then you have the China rose solution, means China rose, the petals of China rose, from that we are extracting the China rose solution. So these are the some commonly used natural indicators. Okay, now if you see the natural, means the natural color, so natural color of the litmus solution is purple, turmeric solution is having yellow color, and beetroot purple again, red cabbage juice is having purple color again, and China rose solution is having the red color there. Okay, now which color they would give in acidic solution and in basic solution, that is how they will indicate whether the given solution is acidic or basic. Okay, now suppose the natural color of litmus solution is purple. Okay, now suppose you have the litmus solution and you put some acid there. You have litmus solution which is purple in color. Are you getting it or not? Let's say here you have in this test tube, you have litmus solution which is purple in color, that you are knowing. Now you put some acid there, so this solution would turn into red. Then if you just put some base there, so this solution would turn into blue. Are you getting? You have this litmus solution. If you put some acid there, it will turn into red color. Originally it was purple color. Okay. Now in next case, if you put some acid there, so the color, the purple color would change into blue color then. Are you getting it or not? So this is how we can indicate whether a given solution is acidic or basic by using this litmus solution indicator. Okay, then we have the next indicator that is commonly used again, that is the turmeric solution. Okay, what is turmeric solution? Originally it is having yellow color. Okay, now if you put some acid there, it would remain same, means the color would not change, it would remain yellow. Now again if you put some base there, so the yellow color would change into deep red color. Okay, that we are using at home, remember this. Next is the beetroot juice. Originally it is having purple color. In acid it would remain same, but in base it would give yellow color. Then you have the red cabbage juice, this one color. In acid it would give pink or red color, sometimes mixed, sometimes red, and in base it would give green and yellow color. Okay, and then you have the China rose solution which is originally the red color. Okay, and in acid if you put some acid there, it would turn into the magenta color. Okay, and if you put some acid there, it would turn into green. Are you getting? So this is how the natural indicators are used, and these are some examples of the natural indicators. Here the most important is the litmus. Okay, the most important one is the litmus. So let's talk in detail about this litmus here. Okay, just note it down before this. Now let's talk about the litmus solution. So litmus solution is a purple dye. It's a dye which is extracted from the lichen which is a plant belonging to the division Thellophyta. Means it's a biological term, Thellophyta. So it belongs to the Thellophyta division of the plants. So that is the lichens. So we extract this litmus solution from the lichen plant, that you have to remember. And this lichen plant belongs to the division Thellophyta, that also you have to remember. Okay, now we have the litmus solution, means in liquid form, it's in the form of dye. Okay, now at the same time we have the litmus in paper form also. You have litmus paper in market. Okay, so litmus is available in paper form also, means it is available in two forms, red litmus paper and the blue litmus paper. Okay, so litmus is available in the form of solution which is purple color, and at the same time litmus is available in paper form also. Means where we have two types of litmus paper, red litmus paper and blue litmus paper, that we are using in our laboratories also. Okay, just note it down, then you discuss about the litmus papers. Okay, so two types of litmus paper are available. Okay, one is red litmus paper, another we have the blue litmus paper. Okay, now let's say we have a solution here, so which is acid. Okay, so here we have acid in a beaker, and then we have a red litmus paper. Let's say this is the red litmus paper, this is red litmus paper, and we dip this red litmus paper in this acidic solution. Let's say this is the acidic solution, and we dip this red litmus paper in this acidic solution. So what would be the color of this red litmus paper? It would remain same, means the red color litmus paper would remain same, means it would remain red. Okay, now if at the same time you have a solution here which is a base, this is a base, in previous one it was acid, here you have a base, and again you put this red litmus paper in this basic solution. This is, we have the red litmus paper. If you put this dip this red litmus paper in the basic solution, so the color would change, the color would turn into blue. Now this red litmus paper would turn into blue color. So this can be asked in the examination also. Okay, what is the effect of base in red litmus paper? So answer would be the base turns red litmus paper into blue. And what is the effect of acid in red litmus paper? So answer would be the acid does not change the color of red litmus paper. Are you getting it? Yes, acid does not change the color of red litmus paper, but the base changes the color of red litmus paper and it turns into blue. The red color would turn into blue. The red litmus paper would turn into blue. Are you getting? This is how we are using the red litmus paper. Now if you have the blue litmus paper, because we have two types of litmus paper. Okay, now let's say we have the blue litmus paper. This is blue. In both the case we have blue. So here we have the first acidic solution. There we dip blue litmus paper. So this blue litmus paper, this acid would change the color of blue litmus paper and it would turn into the red color. This acid would turn the color of the blue litmus paper into red. And in case of base, the base would not change the color of blue litmus paper, it would remain blue. Are you getting this? Very good. This can be asked in the exam. Okay, what is the effect of acid in blue litmus paper? So answer would be the acid turns blue litmus paper into red. And at the same time, what is the effect of base in blue litmus paper? So answer would be the base does not change the color of blue litmus paper. Rather, the color would remain same there. Okay, so this is how today we have read about the natural indicators. So in the next lectures ahead, there we would be discussing about the synthetic and the olfactory indicators. Till then, stay tuned and keep watching. Thank you. Have a good day.